in the waters of the Ganges and the Jamuna rivers in India and marked the antibacterial activity. And he has shown that these are the having the antibacterial action over those type of the bacteria and could pass through a fine porcelain filter. Then later in the year 1915, Frederick Todt isolated these filterable agents capable of destroying the bacterial cultures and producing small cleared areas on the bacterial lawns. So in the year 1917, actually he is the first person, that is the Felix de Herale. So he's the first person to notice this phenomena, postulated that it and he observed the effect was caused by the virus, is not some filterable agents, and it is through the filterable agents only they will eat upon the bacteria, hence they are called as the bacterial eaters, or it is called as the bacteriophages. So its history starts from the 1896 by Hankin, who observed the first keen observation of the Gangas and Jamuna rivers, in which there he found some antibacterial action of the pathogenic organisms. And also it was Frederick also he has shown the culture, some cleared cultures are present, which are called as they are the filterable agents. And it was the first person who has named the bacteriophage by the Felix de Herale, in the year 1970. Are you listening? Hello? Are you hearing?
Yes, sir, you're audible. Can you slide share that? Slideshow.
Slideshow is there. It's not there.
Slides are there, sir. But can you just make that? Yeah, maximize that. Can you just put the slideshow option?
Yeah,
that is done. Yeah. Yeah. Now it's fine, sir. Thank you.
Okay. So therefore, you can see that the Kumbh Mela and everything in India, there are every 12 years there is a Kumbh Mela in Allahabad, Kashi, and Ujjain, like that. There are about crores of the people will take the holy dip in these rivers, and they never suffered with the outbreak of the epidemic such as the cholera, plague, and other dysenteric diseases. So, but and the Frederick thought that Frederick and Hankin, actually it was the first person, Hankin in the year 1896, he observed the Ganga's river's water, and but at the end of this Ganga river, there the outbreak of the people who took the bath and drank this water, they developed the epidemic diseases such as the cholera is there, then dysentery. So all those things have been started, and gastroenteritis. But the people who took the bath in these upper area of the Gangas, they never suffered with these types of outbreaks. Then he told that there are some filterable agents are there which are going to suppress the growth of these pathogenic bacteria. And later it was observed by the Todt, and he said that it is some filterable agents which eats upon the bacteria. And the name has been given by D. Herale, called as the bacteriophage, and they are bacterial eaters. So therefore, and they told that these are nothing but the bacteriophages. We all know that what are the phages there are the bacteriophages. This is the structure is there. The viruses that parasites on the bacteria are called as the bacteriophage, and they are totally distinct from the animals, plants, and other. And these phages will infect only the species specific bacteria only, and even strain specific also. Please remember, it is very useful for the phage therapy that these phages will infect only the species specific bacteria, and not even the other bacteria. For example, you take the staphylococcus will infect only staphylococcus has been infected by the staph phages, and streptococcus has infected by the streptococcal phages, or it is pseudomonas is infected by the pseudomonas phage. So therefore, there are the species specific phages are there which will infect only the particular species of the bacteria. So therefore, this is distinct from the animal and the plant viruses. And these phages are abundant in the organisms on the earth. Even they are present in the sewage water sample. They are present in the ocean water. They are present in the soil sample. So therefore, they are abundant. They are the most populated populations on the earth. Actually, we can say that they are like that. So have either and these phages will have the two type of life cycles are there. So you might have studied in the PVC also, there is a lytic cycle and lysogenic cycles are there. So lytic cycles are very important when the bacteriophage inserts its DNA into the host cell. Then if it integrates, it is called as the lysogenic bacterial life cycle. If it does not integrate with the main chromosome of the bacteria, then this viral DNA freely floats in the cytoplasm, and such a type of bacteria the cycle is called as the lytic cycle. Then then what is the how they are going to infect? So these so you can see here what we are seeing here is, so one can see here these are the bacteriophages which are coming like a helicopter, and they are landing on the bacterial surface. This is we have taken the examples of the staphylococcus. So this staphylococcus on the cell, this virus is coming and it is infecting like an helicopter. And once it comes like a helicopter, and it will lands on that one and it will penetrate. So these phages infect the pathogenic bacteria, and it also can also target the host treatment for the even plants also. So then we can say that in the plants also you can utilize as the plant phages for the biocontrol agent. But here we have utilized this phage therapy for the many type of diseases and diabetic foot diseases, and what you call dysentery, gastroenteritis, and wound infections. For that purpose, we have utilized these phages for the treatment purposes. So this treatment is called as the phage therapy. Then how to isolate these phages? These phages can be isolated by the sewage water. You can take the sewage water. Yes, specifically medical effluent you have to take if you are going to want to really isolate the phage which is responsible for the treatment of the diseases. So are the pathogenic bacteria. So therefore you have to take the medical effluent sewage. The medical effluents, if you have to take it, even though it is there in the sewage also, but it will take longer period for the isolation of the bacteria. So therefore what you have to do means you have to take the little bit of 1:10 volume of the appropriate concentrated broth you have to take. So this lactose broth you have to take, and in which you have to add about 1 ml of the in 10 ml of the LB broth while taking the sample. So therefore, as you know that the viruses are the living entities outside the host. Any host you take, plant host, animal host, human host, or bacterial host, they will live outside the host as a dormant cell. They will be dormant entities. So therefore, they are. That's why we are going to call them as a living entities. We are going to call them as the living entities. And then afterwards, we have to add. Suppose I told you I want to isolate the Staphylococcus aureus bacteria, for which I am seeking the phage. So therefore, first I have to isolate the Staphylococcus aureus only from the pus sample, or from the wound sample, or from the burn sample, or carbuncle, furuncles, whatever the diseases are there, or the pyogenic infections are there. From there, you have to take the sample and you have to make the culture of this organism first from the patient. Then after making the culturing of this organism, we have to incubate this. Then later you have to bring the with the lice with the chloroform and spin for 30 minutes for 500 to 500 RPM for the removal of the debris. Then before that, what you have to do is you have to collect the bacterial effluents, that is the medical effluents you have to collect and incubate along with the SRES. So therefore, this SRES will infect the phages, will infect the SRES, and innumerable number of phages will be released into the bacterial broth. So this broth later it has to be centrifuged. Then we have to remove the debris from the dead bacterial cells. Then we have to filter through the filters. Then it is called as that filtrate contains the filter contains the bacteriophage. So then, therefore, the cultivation and identification involves the first obtaining of the sample either from the medical effluent or it is from the sewage water. Then you have to multiply the phages. So therefore, you have to add the culture in which you are going to seek the phages. So therefore, the solution into liquid media, that is nutrient broth or any triple sugar broth, anything, any broth in which these organisms grow luxuriously. So first, those organisms only have to select. So Staph aureus here I have selected, that is nothing but for the nutrient broth in which the sewage sample is put it. Then after the centrifugation, I have taken and incubated for the 37 degrees Celsius for 24 hours, in which again the increase in the number of the phages will take place because the bacteria has been grown. For example, you take the turbid culture of the Staphylococcus aureus or E. coli, so in which you have to add this filtrate, which have been filtrate which have been from the sewage you have taken. So after 24 hours of incubation, that turbidity must and should clear. That turbidity must and should clear it. So once the turbidity is cleared, so therefore you have to say that we have really isolated the bacteriophages. So therefore, that indicates that, but this itself is not sufficient. So after increasing the numbers by allowing them to infect and reproduce within the fresh host, then you have to separate it. Then this has to be done for the plaque assay. You have to do the plaque assay. So or the spot assay also you can do. So what you have to do means you have to grow the lawn of the Staphylococcus aureus or E. coli, wherein so you have to make the lawn. Then you have to add to the soft agar, you have to add the 1.01 ml of the filtrate of the culture wherein the phages are there, and on the lawn you have to add this semi-solid media. So then you have to incubate as such on the in the incubator at 37 degrees Celsius or 22 for 24 hours of incubation. Then after the incubation, you can observe this type of phages are there. So you can see these cleared area, this whatever the cleared area debris. So this cleared and areas are known for are called as the plaques. These are all called as the plaques, in which it has eaten away the bacterial colony. It has eaten away the bacterial colony in which the millions and billions of the phages are present here. So then this has to be isolated. This has to be re-isolated. This contains only the phages. This wherever the bacterial colonies used to form, there it has not allowed to form the colonies, but it has eaten away the bacterial cell. So on the lawn. So this lawn has been cleared. So therefore this cleared area contains the plaques. This again you have to confirm it by using again you have to pluck this plaque and incubate along in the debris. I mean debris means the turbidity of the Staphylococcus or the E. coli. There if you put it in that, so it has to be cleared. So then you have to see that I have really isolated the plaque or the phages. So in this cleared area, you have to remember that it is nothing but the plaque. That area contains the millions and billions of the bacteria phages, which have been really isolated against the bacterial host. So whatever the bacterial host, it may be E. coli, it may be Staphylococcus, Pseudomonas, or whatever the pathogens you are doing, or against any superbugs. But isolation is very important. The once you have isolated, so then identify the basis is that one viral particle infects only one cell. So many cells may land on the bacterial cell, but many phages may land on the bacterial cell, but only one viral particle will enter into the bacterial host. That even the viral particle means only the DNA of the virus can enter, not and it will remains as a flag on the bacterial cell surface, because bacterial cell wall is a very rigid cell wall. Very rigid, and it is made up of the peptidoglycan, teichoic acid, and other protein peptidoglycans. So therefore, it is very rigid. So for the insertion of its own DNA, viral DNA, it will only insert the bacterial DNA, viral DNA into the bacterial cell. So then this viral DNA decides whether I have to go for the replication or not. The viral DNA, once it enters into the bacterial cytoplasm, it decides that whether it has to go for the replication or not. So this has been controlled. Therefore, the whole bacterial system, that is the biosynthetic machinery, will be diverted and it will ask the whatever the mandate is given by the viral DNA. So because the viral DNA lacks its biosynthetic machinery, so therefore it want to synthesize its own particles or its cell progenies. So therefore it will stop the whole synthesis of whole synthetic machinery of the bacterial cell and starts ask the bacterial DNA to synthesize the required components for this replication of the viral particle. So therefore, therefore this is the various types of phages are there. So as I told that how this cycle will take place, that replication cycles. The first it will have the adsorption will be there. Then most bacteriophages adsorb to the bacterial cell wall. Through some are able to adsorb the flagella or pili. So once they will adsorb or lands like a helicopter, then the penetration is must. Either it is human being, animal being, or plant. The penetration of the viral DNA is most important. So therefore, after the penetration, then the genome of phages which adsorb to the flagella or pili enter through these hollow organelles. Then replication inside the cell will take place by stopping the bacterial metabolic activity to synthesize the phage enzymes. So and the phage enzymes. And after the synthesis, assembling will take place inside the cell itself, inside the bacterial cell. That means it's a head, it's a tail, it's springs, then peplomers, all those things. Totally the mature virion particle will be synthesized in the bacterial host cell itself. Then at the cost of the life of the bacteria, these phages will release into the environment, again in that environment. Or if any organism is there, then again it will infect to the organism. So therefore this phage-coded lysozyme breaks down the bacterial peptidoglycan and causing the osmotic lysis and release of the intact bacteriophages. In turn, these bacteriophages are ready to infect the other pathogens in the cell. So therefore this is a reinfection. So from 50 to 200 phage may be produced per infected bacteria. So from the infected bacteria, that means one single bacteria will produce the 50 to 200 phages depending upon its generation time and other cell wall size. So this is that's why the people will select only the lytic cycle of the bacteria only, because for the fast therapy only the lytic cycle is very important. So this lytic cycle is responsible for the lysis of the bacterial cell, and the lysogeny means the growth suppression will takes place. So therefore the people want to see the lysogeny. Lysogeny means you know that the viral DNA, if it integrates with the host DNA, then it is that cycle is called as the lysogeny. But in any one of the generation, so it may again turn to the lytic, but it is a longer period. So therefore lytic phages are the most suitable candidate for the fast therapy because it lies immediately and decides, and that's why the lytic phages cycle has to be selected. So because they quickly reproduce within and lyse the bacteria in the host range, growing exponentially in number. So therefore these in contrast, temperate phages, the lysogenic phages is also called as temperate phages also, which can bolster their bacterial host virulence, resilience, and general capacity. So are the general unsuitable for the therapeutic uses of the infections for the applications. So this is the cycle. This is just a diagrammatic flow chart of the lytic and lytic life cycle of the bacterial viral particle. So you know that. So here it is coming like a helicopter and landing and inserting it the viral DNA, and it will stops its bacterial DNA metabolic and physiological activities and directs the bacterial DNA to synthesize its viral components. Then it will start the assembling. So once the mature assembling will be there, and this mature assembly will be released into the environment, again it will start for the reinfection. So this is the same thing, lysogenic and lytic cycle. So I told you, so once it is freely, if it integrates with the bacterial host DNA, then it is called as the prophase or temperate phase or the lytic cycle of the bacterial cell. So, but the lysogenic cycle cannot be used for the therapeutic applications. Only the lytic cycles can be utilized for the fast therapy. So, what are the advantages? So, advantages are as we know that the phages are both self-replicating and self-limiting. self-replicating inside the host cells and self-limiting. If and infection also is a self-limiting, because if the same species is not there, it won't infect. So therefore, it limits itself not to attack any other bacteria other than its host. So they continue multiplying and penetrating deeper as long as the local infection is present. So rather than decreasing the rapidly in concentrated below the surface like the antibiotics. So therefore, phages work against both treatable as well as the antibiotic resistant bacteria, and they may be used alone for the alone or with the combination of the antibiotics and other drugs also. And these are multiplied increase in number by themselves during the treatment, or only one dose may be needed also. The dose study will be decided by the doctor. The only slightly disturb the normal good bacteria in the body. Usually they won't harm the microbiome of our system in the intestine, as they are the friendly bacteria are there. And what you call, if E. coli is there, then it little bit harm will take place because it belongs to the members of the Enterobacteriaceae. Otherwise, even it is a strain specific, it is totally species specific in nature. Therefore, other than its host, it never harms the other bacteria. So the phages are natural and easy to find, and they are not harmful to the body, even though they remain in our circulatory system. They are not harmful to our body. They are non-toxic to animals, plants, and the environment. But these are the special advantages of the bacteria, and this can also be used as prophylactically in the hospital environment and hospital sanitization, where the staphylococcus are there or the drug resistant bacteria is there. If you can spray these phages, you can spray these phages in the environment. So therefore, they will attack their host, and they will clean for the sanitization purposes. So these are the special advantages are also there. But what is the problem with this research? Why the people stopped the early research on the phage therapy? As we know that the failure to select the specific phages and target the bacteria in vivo, therefore using them in patients or for environmental decontamination. So at that time, species specific bacteria phages were unable to isolate, and they were difficult to prepare for use in people and animals, and it is not known what dose or amount of phages should be used. At that time, the people never know that how much of the dose to be given to the patient, and it is not known how long phages therapy may take to work. And after that, what happens? And there were we don't have the electron microscopes at that time to study its complete morphological structure, physiological activities of the particular viruses. Only in the 1942, when electron microscope came into existence, then only we can started the research, total research on the virology. So it may be phages may trigger the immune system also. Sometimes it will trigger the immune systems to overreact or cause an immune imbalance. So this is sometimes in some phages it may happens. Some types of phages don't work as well as other kinds to treat the bacterial phages, and therefore then there may not be enough kinds of phages to treat all bacterial infection. So all the bacterial infection cannot be treated by phages. So we have to search for the particular phages for the particular species. So some phages may cause bacteria to become even resistant also. So this is the early problems were there with the research. So therefore, large quantities of the purified phage preparations were not there. Then the filtrations were not there. Then stability of the viability of these phages, how long they will retain their viability, that was also a problem with them. But now most of the problem have been overcome with the development of the electron microscope and lot of equipments which are available for the high-end equipments are available for the phage research. So what I have done in our laboratory, so you can see this is the vector on a mannitol fermentation agar. We have grown the Staphylococcus aureus on the mannitol salt agar. You can see the gram-positive Staphylococcus aureus, and it has been scanning electron microscope. These are the Staphylococcus which are arranged in a grape-like clusters. So this has been used against the MRSA. So then some of them, they have shown the resistance and antimicrobial activity. So this is the sensitive one, and the non-sensitive is also there. So those things are also available. So now you can see that's why against to that phases, what I've shown in the back, this one. So against this phases, these phases, bacterial cell, we have isolated the GRCS phase from our medical effluents from the near MRMC Medical College effluents. So this phase is called as this phase is known as the GRCS. G means Gulbarga, Raju, Chandrakant, Staphylococcus, Plaques. So which have been isolated for the this is actually the new phases which have been isolated against the MRSA, that is methicillin resistant bacteria. Methicillin, you can see the plaques here. That is cleared area is there here. So these cleared area nothing but the GRCS phases. This has been done under the electron microscope, and we have studied that it is belongs to the phase T4 phase group of the bacteria. Then later we have injected to the mice. So you can see here the mice. This mice. So this mice is wanted to use against the diabetic mice. So therefore, by using the streptozotocin, which will injected intravenously to make the animal to become the diabetic in nature. Then later we have added the MRSA, that is the methicillin resistant Staphylococcus aureus. So you can see the infection here. So this is this infection. So it has been after the 2 to 3 days, the pyogenic infection has been started. Then after the pyogenic infection, we have given the treatment by using the GRCS fast. This is a lethal bacteriomic mouse. So it has become lethal. So to this lethal, we have given what you call which we have isolated the GRCS fast. Here, please remember that we have taken the whole phases, not purified phase, the whole phases which have been purified. That is the staph phases only. So that has been given to the animal, and that animal has been recovered after the 2 to 3 days. So depending on the species of the conditions, the parent phase can produce as I told, the 200, and in that, so about it has taken about 3 to 4 days to recover the things. So now we came to know that how to isolate the bacteriophages, how to treat it, and whole phases we have taken. So then what are the phases are there as a human therapeutics available in the market also. So bacteriophages were first used therapeutically in the human during the 1919 to treat the severe cases of the bacterial dysentery caused by the Shigella soni, Shigella flexneri, Shigella boydii. So in the Paris, in France, they have started. All of the treated children were recovered from that, from this type of fatal infection. Therefore, these phages can be given either orally also. It can be given orally, as in the tablet form or in the liquid formulations, thrice a daily after neutralization of the gastric juice with the dihydroxy aluminum sodium carbonate. So therefore, it can be given in the form of tablet or in the form of the liquid preparations. It can be given, and otherwise it can also be given rectally. So because it is 10 ml in twice a daily, as it is going to intestinal diseases, so therefore it has to be given in the 10 ml daily twice. Or it can be applied if any skin infections are there. So as you know that the staphylococcus will also cause the carbuncle, furuncle, and some of the supervity lesions will be caused by the staphylococcus aureus. So therefore, locally also the skin, eye, ear, nasal, mucosa, etc. we can apply over the smear of these phases, and even you can take the whole phases only. So no need to after the purifications. But purifications done only through the filtration. That is a seeds filter or membrane filters can be done from them. So therefore, then after that, so this is the one of the therapeutic. Then what is the comparison between these phases and the antibiotics? You can, as I told, they are very specific and affect the targeted bacteria only. But these antibiotics are non-specific, but even they will affect the normal microflora of the human body. That's why the doctor, along with our antibiotic doses, they will also give this some probiotics also, or some B complex also they are going to give to overcome this type of this one. So development of Phage resistance frequently is associated with the attenuation of the bacterial attenuations, but here antibiotic resistant bacterial strains remains as a pathogenic in nature only. So selecting a new Phage is a rapid process. Production is simple and relatively inexpensive, and developing a new antibiotic is a time consuming process, and which may take several years and be very expensive. So no side effects have been described from the Phages till now, but except that what I told, IgG level increase. So therefore, with the antibiotics, there are multiple side effects have been observed. So the disease causing bacterium has to be identified before the Phage therapy can be started. But here the doctor's antibiotics can be used without knowing the identification of the disease causing bacteria. So therefore, for example, you take the typhoid and malaria always got confused with the doctors. So therefore, if a person is suffering with the typhoid, he will treat for the malarial parasite. So this is the problem with the antibiotics. So therefore, they will give the broad range of the antibiotics. But here it is a species specific, and the disease causing bacteria can be first isolated and later it can be used for the treatment. So therefore, the Phages can be used as a disinfectant, sterilants, biocontrols, and it can be utilized as an antibacterials and applicable on the living tissues. It can also be used for the bioderma like that. So therefore, the patients, then if anybody is suffering with the resistant bacteria, so therefore, what you have to do means you have to go to in the Georgia country, in the earlier Balkan Soviet state. Now it is called as the Georgia, where the capital city is Tbilisi. So there you have to register the patient. Now this is the only the center, and even in France and the USA, Britain. So they are having these treatment facilities is there. So more than 200 diseases can be treated by using the 2000 Phages from the Zagreb. The University of Zagreb is there. So where they are having this type of facilities there. So what you have to do means you have to do the patient register at the Phage therapy center and describe their problem, then send along medical records and bacterial sample, stool, urine, or swab. The sample is tested to see whether the infection can be cured by the antibiotic, or if it is a drug-resistant bacteria. So it can be cured by by the Phage preparation. Once the bacteria is identified and can be cured by the correct Phage, then the center will doctor prescribe the treatment that is prepared in Tbilisi, Georgia, and then shipped to the patient's home. These Phages generally come in liquid form and are applied topically, inserted as drops are taken orally. But in severe cases, you have to go to that country itself and for and it costs about the range of $800 to $6,000 from depending upon the type of diseases. So this is how with the MRSA, this is the methicillin resistant Staph aureus and even Pseudomonas. So for the total 200 diseases, pyogenic infections, skin infections, rectal infections, for that purposes you have to approach them also if you are suffering, if your causative agent is antibiotic resistant in nature. So you can see some of the photographs which have been used by the other members. So here the people, you can see the bacteriophage treatment in 1-year-old baby. So this you can see this is eye infection. So this eye infection, totally pus is there, and this has been given the whole Phages. So for the pyogenic infection. So after 10 to 15 days, so this boy, this infant has been cured. This is totally, you can see the eye has been totally, it is cured here. And same thing, you take the bacteriophages in, this can be used as a wound infection you take. This is the wound infection, and it is a resistant bacterial wound infection with the Staphylococcus aureus. So therefore, they have taken the bioderma. So what you call it as a bioderma. So this is called as the bioderma. So where the smeared Phages are there, and just as an antiplast and what you call, we can call it as an antiplast also. We can call it as an antiplast. So which is the smeared Phages are there. It can be applied over that one. So then later, after 15 days or 7 to 8 days, you can see the curing of the curing of this wound infection. So this is the how the without antibiotics these phages which are can be treated. So this is another research they have done with the Randall Walcott Southwest Regional Wound Care Center, Texas. You can 12th March they have applied over this one. So and 3rd May, because this is a very diabetic foot is so the patient is suffering with the lot of diabetes, high diabetic in nature, and therefore it has taken the longer period to treat the. Then later you can see they are showing the epithelial islands inside the wound. So this is a characteristic of the fast therapy. So that this patient has been cured with the diabetic foot by using the MRSA against the MRSA. So these are the another by using the Pseudomonas aeruginosa. So this is also the same type they have applied over that, and lesion of mice skin infected with the MDR, that is multi-drug resistant Pseudomonas, without without fast therapy. This is the B control. Then that is the A control. So it has been applied over the curing. So you can see the other type of how it is available. This is the fast staph. This is the fast staph. That means Staphylococcus aureus fast is there. It can be given like this. It has been given and treated. This wound infection, everything has been treated by using the fast staph. And so till now I have used only the bacteriophages as a whole. But now we have to think that which is responsible in the bacteria in the viral genome. There is a genome called as the recombinant lysine. So this lysine, so we have in our laboratory we have isolated this genomic DNA was isolated from the bacteriophage GRCS by phenol chloroform method. So wherein we can see the 18 kilobase pair, approximately compared to the molecular weight. So this fast DNA has been taken and it has been digested with the sequenced at the Maryland University. It has been sent to them where I have been gone there for my higher research training there. And there we have seen that there are the two types of enzymes were obtained. One is the putative the endolysin, another one is the putative amidase. So however, the endolysin has been given a lot of impression that can be cured, and it has been cloned. It has been cloned in the E. coli. The MECA gene has been cloned. So this is the putative amidase is there. Then for the putative amidase, it has been, but this has not given much result. This endolysin, which is the genomic DNA, is there from the virus. So GRCS fast, it has given a lot of improvement. About 98% of the successful recombinant lysine has been obtained after the cloning. So this cloning, the part of work has been done in the Niemann's with the help of the Yogeshwar, Dr. Yogeshwar there, and they have given the chance to clone the cells in the E. coli. So, but putative amidase, that enzyme has not given the very successful result, but endolysin has been given a successful result. So this is the publication of the sequence of the GRCS fast I have done at along with the Daniel Nelson, Steve Smith, and we have provided this GRCS fast to them as it was costly at that time. So this cloning of endolysin from GRCS, as I told, it has been see, you can see it has been used with the PBAD 24 plasmid for in the E. coli DH alpha 5, and it yielded a very good result. You can see that the bands also the same bands. So where the 28 kilodaltons were there, SDS fast, and after the purification, so this class is called as the Plaque class, Plaque GRCS. So this is the Plaque GRCS endolysin protein was purified by affinity column chromatography, and now we are going to use this one for our next research. So you can see that the graph we have put up here is where the endolysin has completely treated. Endolysin has completely treated. There is nothing but the Plaque GRCS fast. So the name has been given for this recombinant lysine is the Plaque GRCS fast. So this GRCS fast has been utilized for the treatment of the turbidity assay of the Staphylococcus aureus or the MRSA, VRSA, vancomycin resistant Staphylococcus aureus. So almost all the resistant Staphylococcus aureus has been treated by this recombinant lysine. So this is the spot assay of the agar well diffusion method. So where I have added the against the staph aureus. So you can see the complete the susceptibility of the spot test assay of the different concentration of the Plaque GRCS phase. So therefore this phases, the putative amidase as well as the GRCS Plaque GRCS 100 microgram per ml has been utilized. So this is the Endolysin spectrum of lytic activity. And this is the total my research in my laboratory. This is the Staphylococcus aureus. These are the phases where these are the genes, MECA gene, FEMA gene has been utilized for our laboratory. So this is the person who have worked on the whole genome, whole phases of the GRCS phase. This person now is working as a scientist at the Bharat Biotech for the nasal corona vaccine, and this boy has been appointed. This boy, Dr. Manjunath, so he has been appointed at the Hassan Medical College, and who worked on the recombinant glycine of the GRCS phase. And this another person, Rahul, Dr. Rahul, who worked as worked on the recombinant glycine against the different pathogenic bacteria, and this person is known now. He has joined the Karnataka State Police Department as a scientist in the forensic science, and this person is in the Davangere University, and this is in Ballari University. So therefore, these are the total my students' work, and they have done good, excellent work. And this is the bacteriophage therapy and its applications for the resistant bacteria. Thank you very much, and I thank Dr. Shubha and principal and the staff members for inviting me for this international webinar. Thank you. Over to Shubha, madam.
Thank you, sir, for giving excellent coverage on speech. Participants, if you have any doubts, please put it in the chat box.
Let them know that already there are very informative and excellent session.
I think if any questions are there, then we will, or they can ask through my email also. If you are having questions, you can ask.
Can you share the PPT through email?
Sir, can you please share the PPT through email?
Yeah, I can share it.
Thank you, sir.
Send to madam.
It will be helpful for the participants.
Yeah, I will send it.
Sir, there is one question. How is phage therapy different from antibiotic therapy?
Antibiotic, I told you, the antibiotic is also a good one, but if the causative agent is resistant, and the doctors cannot treat the patient. If so, for example, I told the last antibiotic of choice is vancomycin for the Staphylococcus aureus. So therefore, in such cases, the patient will develop the abscess, and the whole body will become the pyogenically infected. So in such cases, this can be used as an alternative therapy. It can be used as an alternative therapy as the whole phases, or even the recombinant glycine can be utilized.
Thank you, sir. Sir, another question from Ashwath. Sir, is any chance for the recombination of the bacterial virulent gene to phage progeny?
But the phage progeny is not there. But from the phage progeny only can be cloned in the bacterial cell. So therefore, they will produce the recombinant glycine, not from the bacteria. The phage only, the phage DNA only has to be taken, and it can be cloned in the E. coli, as we have done the Plaque GRCS recombinant glycine.
Thank you, sir. And one more question is there, sir. Sir, what about dosage for particular disease?
Yeah, that will be decided by the doctors. Then then after once the animal experiment is completed, then human experiment trials, then due to the LD50 and other things, so that is lethal doses. The dose will be decided by the doctor.
Thank you, sir. There are a lot of appreciating comments in the chat box, sir, saying the session was very informative and knowledgeable. Thank you, sir, for such a wonderful session.
Okay, madam. Thank you very much. Thank you, Shubha madam and principal and all the participant for giving in a perspective. Thank you.
Now we are at time.
Now we are at the end of the session. No event can be successful without people who dedicate their resources and time to make sure everything is faultless. Now I would like to thank our speaker, Dr. Umesh sir, who has delivered lecture